Okay, so let's now take a look at lipid nomenclature. There are specific rules that are in place for describing lipids, and so that from the name of the lipid we can actually draw the structure, and vice versa. Once we know the structure, we can draw the name. So the basic structure of a fatty acid is shown here. And a fatty acid, because it's an acid, consists of a carboxylic acid at one end. You'll notice that there is a methyl group at the other end, and a long hydrocarbon chain connecting the two. This particular fatty acid has 16 carbons to it. 16 carbons. The carbons are all linked together with single bonds, and so this is the fatty acid that we can make with the fatty acid synthase complex. Because it's 16 carbons long, we designate its name as C16. Because these are all single bonds, there are no double bonds in here. This is a fully saturated molecule. Saturated meaning no double bonds. We designate this as 16 with a colon and 0. 0 indicating no double bonds. This particular fatty acid is called palmitate, and this is the most abundant fatty acids that we have. Now, we also have a numbering system and a lettering system. The number 1 carbon is the carbon of the carboxylic acid. So this would be essentially listed as carbon number 1, 2, 3, 4, etc. The number 2 carbon is also termed alpha. The number 3 carbon is beta. The methyl carbon on the end is the end of the fatty acid and termed the omega carbon. Now, if we introduce a double bond somewhere in this fatty acid, we can do that by using a desaturase enzyme. A desaturase enzyme is designed to introduce double bonds. So let's say, for example, we want to introduce a double bond at carbon number 9. 1, 2, 3, 4, 5, 6, 7, 8, 9. So this particular bond here between these carbons, carbon number 9 and carbon number 10, would be processed by the desaturase enzyme to create a double bond. So this bond here would be oxidized. The name of the fatty acid changes. It now becomes 16 colon 1 delta 9. And this indicates that we have a 16 carbon fatty acid. We have one unsaturated bond, one double bond, and that is at position of carbon number 9. Now, something to note, that humans can introduce double bonds at carbons that are less than or equal to carbon number 9. Any carbons, any fatty acids that have double bonds beyond number 9 are from a plant source. So carbons that are greater than number 9 are a plant source. Now, if we have two double bonds in here, let's say that we have one here at number 9, 10, 11, 12. Let's say we introduce one here at carbon 12. The nomenclature would then be C16 colon 2 for two double bonds, delta 9, comma 12. And this would indicate 16 carbons, two double bonds at positions 9 and 12. So this particular structure is a monounsaturated fatty acid. This structure is a polyunsaturated. Another important feature of double bonds is that they can be in either a cis or trans configuration. The double bonds that we introduce using a desaturase enzyme are all cis bonds. We cannot introduce trans bonds. Trans is a commercial process of partially hydrogenating a polyunsaturated fat, and we'll be able to introduce trans bonds. The difference between the two is a cis bond, recall, puts a kink in the fatty acid. So if this is our carboxylic acid end, this is our omega end with a methyl group. If we introduce one double bond by the desaturase enzyme, this will create a cis bond and create a kink that will be bent. The trans enzymes, recall, will look like this. This would be a trans bond. CH3 at one end, the omega end, and our carboxylic acid functional group at the other. Now, it's these two structural differences, these two different structures, that lend to the problem with trans fatty acids. So these are trans fatty acids from commercial sources. The benefit of this, of a trans fatty acid, is that chemically it increases shelf life of food. The food does not go bad as quickly as with cis bonds, and so therefore increased shelf life, increased longevity of the product in the market. The problem with trans bonds is that if you have these double bonds that are in this configuration, and you have a lot of them around, such as in processed foods, we can form between these bonds interactions that interact between the hydrocarbon chains. So these are hydrophobic bonds. Because there are so many that can form, because these structures can approach very close, the strength of these bonds is dramatically increased. Now compare this with cis bonds. We might potentially have a cis bond here, and say, for example, we have a saturated fatty acid with no double bonds next to it. In reality, this is what our fats look like from endogenous synthesis. Now notice the bonds that can form hydrophobic bonds, distance matters, so only when these hydrocarbon chains are close together can the hydrophobic bond form with any type of strength. So these hydrophobic bonds have a decreased strength. In other words, these are weak interactions. Now when we think about cardiovascular disease, specifically atherosclerosis, this is where trans bonds cause a problem. Now we know that atherosclerosis is primarily due to the oxidation of LDL lipid particles, which are mostly cholesterol. But there are small amounts of triglyceride. And these triglycerides can have essentially, from dietary sources because of processing, they can have trans fatty acids connected to them. If we take these trans fatty acids, mix them with the hydrophobic molecule of cholesterol and oxidize LDL, and all of this is taken up by a macrophage, this will lead to accelerated foam cell production. Conversion of the macrophage to the foam cell because of the excess cholesterol and fatty acids that are taken up. Now the problem is resolving this. The question is if we have these very highly hydrophobic interactions between these trans bonds, do we have a mechanism in order to clean this up, essentially for the macrophage to be able to break these hydrophobic bonds and get rid of these excess trans fatty acid? The answer to this is no. Because of the energy that is required to break the trans fatty acids interactions, we have no biochemical mechanism to break these bonds, because it's going to require a large amount of energy to pull these structures apart. Hence, over time, these trans fats, if they continue in the diet, will increase in their accumulation. And this will aggravate the formation of an atherosclerotic plaque. Now, the other part of this we can consider, and this is a new theory in obesity, is that if you have lots of trans fatty acids that are stored as triglyceride in fat tissue, in order to access these fatty acids and release these fatty acids, recall we have to have lipase activity. The lipase activity is going to be a hydrolytic reaction to break the triglyceride into fatty acids and eventually its glycerol backbone. Now, to remove the triglyceride from the lipid droplet, we have to apply energy. There are mechanisms in place in order to pull the triglyceride out of the lipid environment of the lipid droplet so that the lipase can perform a hydrolytic reaction. So it has to add water across there to break that ester linkage. If the fatty acid that is attached to the triglyceride structure is of a trans bond, the interaction between these trans structures being linear is going to be dramatically increased. So that means that the fatty acids are forming greater hydrophobic bonds, and it's going to be very difficult to pull that triglyceride structure out so that the lipase can have access. So the thought is, with increased consumption of trans fatty acids, the mobilization of fatty acids during fasting is decreased simply because of this strong hydrophobic interaction. So again, over time, with larger consumption of trans fatty acids, it becomes increasingly difficult to remove these fatty acids from the lipid stores that have created obesity, and that will aggravate and accentuate the problem.